All right, well, today I just want to tell you a little bit about how we're using the FlowCAM to do some plankton studies in San Francisco estuary. And, of course, I'm Peggy Lehman, and I work for the Department of Water Resources. Today I thought I'd tell you a little bit about what the FlowCAM is, give you a brief description, a little bit of how it works and some of the data that we get from it. I'm going to tell you a little bit about how we use it to do phytoplankton, zooplankton, and Microcystis studies. I'll show you some of the products that we've come up with, make a little bit of an evaluation and some recommendations on how to use it, a brief summary, and then I'll allow some time for questions. And altogether, that should be about 45 minutes. So what is a FlowCAM? A FlowCAM is technically a digital imaging flow cytometer type system. It's comprised of a computer and a spreadsheet program, as you can see in this top piece here. The guts of the machine is really a microscope, a flow cytometer, a laser fluorescence probe, and a digital camera. This is made by a company called Fluid Imaging Technologies Incorporated. What I'm showing you here is the portable model. There is also a desktop model. We take this instrument right out into the field with us on the boats. It's about 80 pounds, and it's on a roller, so it's pretty convenient. We look inside the machine to see the pieces that go into this. It has quite a few components. This section here is our sample port. This is where we put our sample in. You can put it in just straight from a pour in, or you can have a feed from a line straight into the water. But the sample goes here through a glass cell, and then there's a microscope system that sees the particles that you have in the water. It has a magnification anywhere from 2x to 20x, so you can see quite a bit. As the samples go through, there's a camera that takes digital images of the particles. And then, depending upon your use, you can also add a laser system to this. We use a fluorescence laser so that we can identify chlorophyll in the phytoplankton cells, so that every time a phytoplankton cell with active chlorophyll comes through, the camera will take a picture. This is linked up to a pumping system here that brings the sample through, and that's the flow cytometer component of it. All these cells are moving in a fluid stream. You can change the image in terms of its clarity by this little knob here, and so it's a pretty nice little system. So what does this FlowCAM do? Well, as the samples go through this little cell, it will take a picture of the objects in the water. It'll measure the dimensions of those images or objects, the volume of them, and it'll count the number of the objects. As it goes through, it's going to track the volume of the sample. If you develop a library of your images, it'll automatically classify the objects into groups, like taxa. And because you're collecting the volume, you can compute the abundance, volume, and size of each object per unit volume. So it's a pretty neat little system there. And all that information is then exported into an Excel spreadsheet so that you can manipulate the data. Now, the FlowCAM's a pretty intricate little machine, and rather than try and go through the details myself, I pulled off a video from Fluid Imaging Technologies of how they actually operate the machine. And what you see here is the operation of the laptop version. So I'll run this. It takes about a couple minutes, and let me know if that goes well.
This short video will show how easy it is to use the FlowCAM to rapidly acquire large sets of particle image data for analysis. First, the operator merely pipettes the sample into the sample funnel. At this point, the run is invoked from the Visual Spreadsheet software. The software-controlled syringe pump automatically primes the system, pulling the sample down through the flow cell, through the pump, and into the discharge container. This animation shows the sample being pulled through the flow cell in the optical path. As the sample passes, a flash LED is triggered behind the flow cell, which backlights the sample and effectively freezes the particles in motion. Simultaneous to the flash, the computer instructs the camera, viewing the sample through the microscope optics, to acquire a frame of the flow cell view illuminated by the flash. When each frame reaches the computer, the software automatically extracts separate images of each particle or cell for further processing. This screen capture shows what is actually seen by the operator during acquisition. The upper right window shows the actual camera field of view of the flow cell. The lower right window shows the actual particle images as they are segmented out of the full field camera image. Each particle image is stored in what is called a collage file, and up to 32 different measurements are stored for each one of the particles. If you look closely, you can see small red boxes in the live video window representing the area being cut out for each particle image. You can see how rapidly the images are acquired. The operator is pointing to the live video window. Now he's pointing to the software's main window, where graphs and summary statistics for the particle measurements are updated and displayed in real time during the acquisition. And here you see the run is complete, and that's how easy it is to acquire data with FlowCAM.
Well, I hope that gave you an idea of how the machine actually works. What we do is we use the FlowCAM to do a lot of plankton studies in the estuary, and we do a lot of that on the water. We take the FlowCAM and we strap it down to the boat, pad it with pillows and such to make a stable environment, and with that, we can go through some pretty rough water and difficult conditions without any problem in the operation. You can see here that we're out near Antioch Bridge and ready for a long day. When we run these samples, we get these kinds of images of the delta plankton. These are plankton that come right out of the water. We run them for about 6 minutes and get about anywhere up to 700 cells displayed. And these files are digital files then that we take back with us to the office and then process there. But we have the image set for the live phytoplankton that we see in the water. Sometimes we can't do the phytoplankton live, so we'll do preserved samples. We take samples from the water, preserve them in leucols, and then bring them back and process everything at the lab. And this is what that would look like. The difference between the two is really that our delta has a lot of detrital material. And so when we go to process the samples from a preserved point of view, there's a lot of detritus in it. In the live processing, we can actually use a fluorescence probe to select live phytoplankton, so we don't have all the detritus in there. The detritus samples have up to 2,000 or 3,000 images, so we need to sort through that to get the phytoplankton. But it's not difficult; it just takes a little more time. So we prefer to use the live system so that we don't have to sort through this detritus, and that's just pretty much in nature of the San Francisco estuary that's so turbid. It depends upon your system how much extra material you might get and what kind of probes that you can use. One of our success stories in using the FlowCAM has been to do Microcystis sampling. Microcystis is a cyanobacteria that came into our estuary about 2000. It forms these really thick layers in the water column. It's difficult to sample because it has a size range from 6 to about 50,000 micrometers. And because it has this, as you can see on the right, kind of this little tissue kind of form, it in preserved samples, it balls up and it makes it very hard for the person to identify them well and to get the volume of them very well, or even count or estimate the counts for these. Instead, we use the FlowCAM because it's in the sample is coming in through a flow stream. These colonies, which are flat in shape, kind of splay out and lay out so that we can see the entire colony in a flat shape, which gives us the ability to do a really nice estimate of volume and subsequently abundance. So we can see these individual little cells, which are like 4 to 6 micrometers in diameter, all the way up to these very large colonies of over 300 micrometers in size. We use different magnifications to get at these. As I said, the FlowCAM will give us anywhere from 2x to 20x, so we're able to see the whole range of these, and so it's been a very good use for a Microcystis colony identification. And when we look at the microscope evaluations, we find that we get a correlation of almost 0.9. So we were very pleased to find this way of looking at the Microcystis. It's very fast, easy to do, and very effective. We've also been using the FlowCAM to do laboratory analysis of zooplankton. Again, we find this very, very effective. We use a larger flow cell that we would use for the phytoplankton, but we're able to see them all very clearly. As you can see here, we have adult copepods in the sample. We see their juveniles, the nauplii. They all come in very nicely and are quite distinct from the background material. We think that we can do a good job looking at the zooplankton taxa here in terms of, say copepods versus codocerans, that sort of thing. It's more difficult to split out some of the adult zooplankton if they have a similar look. You never quite know how the zooplankton are going to come through in the system. They could be, up or down or sideways or folded a little bit, and often the identification for these guys comes from some small little CT on the side of their body. And if you're lucky enough to get a good image of those, you can get it down to species. If not, the general taxa are quite simple to do. A lot of people will adjust by taking a small subsample, taking a look at it under a dissecting scope, and then by that they know what the general species are. So there are ways to get right down to species for some of these organisms. In terms of the analysis of the data, again, I think this is a great time to show you a little bit how the FlowCAM actually can be used to separate out the various objects in the sample that you have. A lot of it's somewhat automatic, or you can make it somewhat automatic. And again, I think fluid imaging has a very nice video that addresses that. So I'm going to play again another minute or so video for you to take a look at how that's done.
This movie will show you how visual spreadsheet can be used to do automated classification. That is, to automatically define particles based upon what they look like. This particular example is one of water. You can see all kinds of different types of algae are being captured in the water sample as we're running it through the FlowCAM. This particular application actually uses the fluorescence trigger in order to trigger the camera when something that has fluorescence in it passes by. Once the run is finished, we can use the interactive scattergram to select all particles, and then we can flip through them using the arrow buttons up at the top of this window. The next step is to open up a classification window by opening classification window. At this point, we're going to bring up a template that we've already built with certain types of algae in them. We now define the output classification, and now you can see there are tabs set up for each of the types of algae we're going to be looking for. Each category of algae was predefined by selecting organisms and storing them in a library. At this point, we just run the classification, and every one of the images is divided into these classifications. These are the Anabina. Scroll through a few of those. Here are the Cosmarium, and finally the Storastrum. It's that easy. Last but not least, I'll export the classification data to a spreadsheet so that I can see the results, and then I can export them to some kind of a report. This brief video
So here is an example, a close-up example of some of the summary data that you can get. You have about 30 different variables that you can ask the FlowCAM to give you, such things as aspect ratio or diameter or volume, etc. And it will summarize all your particles in that way. Or you can ask it to summarize the particles based upon the groups that you set up. That might be genera or species or large taxa. And all that is based upon the individual record for each particle that's in your sample. And you can see each one of those particles and the data associated with it in the Excel spreadsheet. So it gives you a lot of information to work with in evaluating your sample for whatever you want it to do. Well, I want to tell you a little bit how we use these data. We've been doing a lot of high-frequency studies in the Delta. This map here shows you a picture of the Sacramento-San Joaquin Delta region. This is the Sacramento River here and the San Joaquin River here, and they merge and then move into northern San Francisco Bay. This is the Sud Bay right here, and then down to the main bay. Over time in the Delta, we've seen a decline in the productivity of the fishery, and one of our concerns has been whether we really understand the nature of that decline. We sample because of restrictions and time and money, et cetera, at only a few stations. We don't really know what's happening along the full course of the river. But with these things such as the FlowCAM and other instruments I'll tell you about, we're able to do a high-frequency analysis, giving us an idea of really how the productivity in the system is changing throughout the system and in those stations where we don't normally collect data. So what we do is we link the FlowCAM data, which we collect at about every 3 kilometers for this study, which is about every 10 minutes we're sampling. We link that up with continuous readouts through the YSI instrumentation for water temperature and specific conductance, et cetera. And then we link that up to phytoflash fluorometry. We're looking at the health of the phytoplankton, its photosynthetic efficiency, and a GPS unit so that we can link all this together to get a continuous set of information along these river courses. And because of the FlowCAM, we're able to come up with a whole lot of information about how the phytoplankton, in this case, vary along the river course. This is about a 90-kilometer stretch that we're working through here. And, for instance, the first thing we look at is things like abundance. We've got, like, total abundance, and then the abundance of the individual genera that we have. If you look at May, for instance, we see that the total abundance of diatoms, these green bars, really drove the abundance in the phytoplankton community. We look at August, we see there that the total abundance was primarily driven by cryptophytes. And you can see how there are a lot of changes that occur as you go down the river at every 3 kilometers. It's quite a bit of variation and something that we didn't realize until we saw these images. We can also take a look at genera as we go along the river at high frequency, and again, these are diatom genera. We can see that there is an enormous amount of variation as you go along the river. There are a lot of hot spots where you see various groups or genera occur. It is not an evenly based system. We have a lot of variation, and that variation affects a lot of things, such as the size structure and the biomass of the individual cells available to the zooplankton. We also look at community composition as a percent composition. You can see here when you're looking at such high frequency, you get a really good picture. This is a diatom bloom that occurred in May of 2010. You can see the diatoms are really abundant throughout most of this stretch of 90 kilometers, but then as you start to get toward the end of the stretch, up near Rio Vista, you start picking up a lot of cryptomonad types. So it's a very, very complete picture of how things have changed and how we might expect them to affect the fishery production. One of the nice things about the FlowCAM is that it gives you an estimate of the biovolume of the phytoplankton. And with that, we can do a quick calculation to get the amount of carbon that each cell has, and of course then the total carbon for the sample or the total carbon for a piece of the object set, such as say diatoms or cyanobacteria, et cetera, and that's what this shows here. So we can take a look at the total biomass on this black line, for instance, in May, and we can see that in May most of the biomass was controlled by diatoms. Again, I'm going to direct you to August, where as before we had most of the abundance in August was due to these cryptomonads. You see here when you look at the total biomass, though, there's quite a bit of it due to cryptomonads. On the other hand, if you look at the biomass, a lot of it is due to this red color, which is the cyanobacteria. So a very different picture of the system when you look at it in terms of abundance and biomass, and with the FlowCAM, the biomass is computed automatically. And really good for us is the ability to split out the sizes of the phytoplankton. The FlowCAM gives you a length, width of every object that's in there. And so with that, we can determine the overall size structure. These yellow lines are abundance, and each one of these colors represents a size range. So the very dark color here is less than 20 micrometers, and the very white color is greater than 100 micrometers. So you can see again, as you go along a 90-kilometer stretch of the river, that there are huge differences in the size structure of the phytoplankton community, and that is of immense importance to the zooplankton that have specific size structures of which they feed on optimally. It's just quite an image of the system that we didn't really understand until we got these pictures. Of course, one of the things that I like to do is to look at these biological gradients and match them with environmental gradients in order to adjust the ecosystem so we can enhance production. We really need to understand the relationship between the environmental variables and the biological ones. So right here, I'm going to give you an example of how that works very nicely. With these high-frequency data, we can do a really nice distribution along the longitudinal gradient. That's longitude along the x-axis here, and this is month along the y-axis. This is May through October of 2010, the Sacramento River. These data represent percent diatom biomass, and you can see that in May, that diatoms are very abundant throughout most of this 90-kilometer reach. They were also abundant in the first portion of that reach in September, but not abundant at all in the July period. When we look at water temperature as one of the variables that are important for diatoms, you can see that those diatoms in May corresponded to about temperatures of around 18 degrees. In September, again, in that low end range, about 18, 19 degrees. In July, when we didn't see any diatoms really, what we saw was a very high temperature spike, somewhere in the 23 to 21 degree temperature range, temperature ranges that are not beneficial to diatoms. This is a very classic response of diatoms, that they occur at lower temperatures, and here you can see it very clearly when you have these high frequency images. This is something we couldn't do with the standard stations that we sample at low frequency. Next, I'd like to give you a little bit of evaluation of the FlowCAM to help you think about it. It really has a lot of advantages, for us at least. It's non-destructive. It doesn't destroy your sample in any way. It handles a wide range of object sizes, so anything from 2 micrometers on up is pretty much in the size range for the FlowCAM. It's very simple to operate. We have people running this within a couple hours of introduction to the machine. I have people that are high school students running it. It doesn't take anyone who's quite expert on it just to run it. It's much easier to use than the microscope for identification because you have an image set that you can work from. People find themselves transfixed by the images, and they stare at them for hours. It's a lot of fun for them to see these. It avoids all the problems of focusing with the microscope in those long hours of highlight, which are very difficult on a person to do. It's really reliable. We don't find any problems with it. We've done quite a bit to the machine in the process of sampling, and it just seems to kind of come along and work really well. It's very fast processing time because your images are what they are, and so you just go right through them really quickly. You have the libraries to help you out. That automatic sorting feature is really nice to help you get through the simple sets. We find very good precision with it. Pretty much it's pretty spot on in terms of replication. One of the things that we were concerned about with the FlowCAM was that in San Francisco estuary we would have a problem with turbidity, but we didn't have any at all. Our system was one of the most turbid estuaries in the world, and yet we had no problem at all seeing these images, so we were really happy with that. And most images are very sharp. Once you set your image focusing up, they're very sharp. And the nice thing about this is that you get a permanent record. Those images are there in digital format. You can look at them and identify them one day, and if you find that you feel you identified an organism incorrectly, you can go back and identify it in a different way. And you can do that throughout decades because it's always there. And with the Excel spreadsheet record, it's very simple to make adjustments to the data set. And relatively, we think it's inexpensive. We put a person there, like a scientific aide, to run all these samples through, and that's pretty quick. Normally, you send out to a lab, it's $200, $300 dollars per sample, so we feel it's pretty cost-effective for us. There are some disadvantages. It has a relatively high initial cost. I think the last machine I bought was something like $86,000 or so. Depends how many samples you're doing. If you're doing a lot of samples, the cost-benefit starts to become more positive as you go along. There's decreased resolution below about 10 micrometers. The machine is apparently works around 2 to 3 micrometers. But we have a lot of plankton often that occur around 1 micrometer in size. So those picoplankton are harder to see. They keep improving the machine, and I can say now they're down to about 2 micrometers, so they're doing pretty good. You have a range of 2x to 20x, so you can see quite a bit. But if you want picoplankton, then you'd have to do that sort of another way at this time. We do find that when we're working with preserved phytoplankton samples in the San Francisco estuary, that the high number of detritus objects in there get in the way of processing. It's just more tedious because you get a couple thousand images when you have detritus versus some hundreds with the phytoplankton. So it's more tedious. It's not impossible, just a little more tedious. So we prefer a live sorting as much as possible. Again, species identification can be limited. The cell has water flowing through it, and it brings the objects through with it, and sometimes it's very close to the microscope ocular or far away, or can be tilted or whatever, and so you can't always get the best view of the cell you're looking at. So species identification is limited. It depends what kind of cell you're looking at. Some people get around this by taking a little subset and checking on the species if they have a question. So that's variable. We feel very confident, though, that for larger taxa and genus, for phytoplankton, for instance, that we're really good. And species just depends upon whether or not we can know what it is and from this sample or from ID sample. The field use is really good. We use it in the field, and it gets through some pretty nasty environmental conditions, but it does have to be fairly well controlled. You can't take it on a whale or go bumping along the waves. It is a microscope. It does have a glass cell that it works with, so there has to be some controlled environment, a padded system, and some temperatures below 100 work best. And even though there's a lot about this machine that is very automated and all, we do suggest that if you want to use it, you have to have some kind of dedicated staff, someone who really understands the phytoplankton or zooplankton, whatever you're doing, and knows how to identify them and can determine when the machine is making accurate sorts or not. No machine is going to perfectly identify the phytoplankton, and given the various shapes things come in, nothing will be perfect. So there has to be a dedicated staff to understand the system and use it properly. With that, though, I can make some pretty good recommendations. For the phytoplankton, I think that here in the San Francisco estuary, it's really best for live phytoplankton collection. It's fast, efficient. We feel real good with those samples. Very good separation to taxa and genera. You do need some expertise to develop the libraries and check the identifications, but that's not too difficult. Very fast and easy to process samples. We can do quite a few in a day. It just depends upon how much time you want to spend for each sample. In the field, we're doing these high-frequency studies. We do runs for about 6 to 10 minutes, and that's our sample. For Microcystis, this is really a great system for Microcystis. It's very hard to look at Microcystis under a microscope and preserve samples. It balls up. It's very hard to get an estimate of biomass of a microscope. But with the FlowCAM, just the way it lays out in the flow stream, gets a really, really nice estimate of biomass. It's very fast. Doesn't require anyone with much expertise to work it because the cells are so distinctive. So it's really, really good for Microcystis identification and biomass estimates. Again, for zooplankton, it's very, very good. Separation to taxa is excellent, and you don't need a whole lot of expertise to get this. We do carbon estimates for zooplankton very rapidly, and we're very pleased with what we're able to do. As I say, we have people from high school students on doing these samples, and they do just great within a very short time. So in general, I think it's pretty good. I think, that I can say that I believe it's, so far for us in this system, it's been fast and easy and cost-effective. We can identify phytoplankton, Microcystis, and zooplankton. We're happy with the identifications to genera or larger taxa. But I do want to, make sure that everyone knows that despite all the automated features, that you do have to have some dedicated staff that are knowledgeable about the kinds of organisms that you're trying to look at and keeps an eye on the machine and how it's operated. There's a lot of information out there. You can go to fluid imaging technologies. On their website, they have a whole lot of white papers, some more videos, and a lot of information, and some technical people that can help you if you have a lot more questions. And with that, if you have any questions of me, I'd be happy to answer those at this time.
Hi, Peggy. This is Ted. How's been your experience with collecting samples and then presenting them fresh to the FlowCAM within a few hours? Say, if you take it, you can't take it out in a small setting because you don't have the boat support for it, but you could take the FlowCAM to a lab nearby and then bring samples right away. What have you had experience with that, and how well has that worked?
Yeah, we haven't done that. We take the FlowCAM right onto the water so the samples are fresh. The problem, of course, is when you have a live sample, you can have live zooplankton in that sample too, and so they can eat your sample as you wait. So
right.
I don't advise it. I think it should be possible, and I haven't done it, of course. I think it should be possible to take a fresh sample, chill it, so that you reduce the activity of zooplankton or any other carnivorous phytoplankton, such as dinoflagellates. And then within a short period of time, an hour or so, still run your sample and be fairly confident you have a good sample. But as I said, I haven't tested that.
Question, this is Lillian. Can you hear me?
Yes, thanks, Lillian.
Yeah, this is Lillian, San Diego Water Board. I have two questions. One is, did you try out different preservation solutions like lugols and formalin? And then the second question I have, how much, do you have like a good cost estimate how much the maintenance is per year? You know, do you need to buy new of these flow cells or these glass cells? So what is the, overall, is it pretty low, the overall maintenance?
Okay, so the first question was, have we tried other preservatives? We use lugols for preserved phytoplankton. That's pretty much the standard material you use. You wouldn't use formalin for phytoplankton. For zooplankton, we have used a formalin-type preservative, but we strain that off and shift it out to water before we run it through the FlowCAM, mostly because that's a safety factor.
Okay.
Let's see, what else have we used? We use ethanol a lot to preserve our zooplankton. Again, we wash it off before we use it. The system itself consists of a little plastic or Teflon tube, goes into a glass flow cell. And so anything that would corrode the Teflon tube, or maybe it's Nalgene, I'm not quite sure what it's made of, it's plastic. Whatever that's made of, whatever would corrode that would break down over time. So water is a pretty good thing. It's not going to damage that. But anything that would go through that would work. The issue would be primarily how much you want to expose staff to it. So, for instance, at one point we did use a modified formalin solution for the phytoplankton. And when we did that, we had to put the FlowCAM in the fume hood so that we wouldn't have a hazardous condition for the staff. So I guess the point is you can use anything pretty much as long as you're willing to take the degradation to the tubing. You're willing to put it in a environment which is appropriate for the chemical.
Okay, thank you.
And in terms of cost, in terms of maintenance, it depends. If you're a klutz like myself, I break these 100 microliter flow cells somewhat. We get a lot of use out of them. We'll get a whole season's use out of them. I guess, the flow cells, I forget what we last bought, maybe 3 or 6 of them or something for a few hundred. That's about all you really need, those flow cells. It depends upon whether or not, you use them a lot and break them a lot. They are somewhat delicate, not impossibly delicate, but they are somewhat delicate. There are different versions you can obtain too that are less delicate, and we're working on some of that. But there isn't a lot of maintenance necessary at all. We have some flow cells we've used for a number of years now. On a yearly basis, the only thing you'd have to do is make sure you had enough flow cells to work with. Yeah. Mm.
So overall, the costs are pretty low.
Yeah. Yeah.
The maintenance costs.
Right.
So it's the one-time equipment that's expensive, but the overall yearly maintenance is not that high.
No. When you want to upgrade, though, which we are doing continuously, then it gets more expensive. So we're shifting from, say, XP to Windows 7. Then you're talking about a few thousand. But considering the overall cost, we didn't think that was too bad. So.
Thank you.
Yeah.
This is Karen Taberski from the Water Board. I have a question. Peggy, it seems like you usually take discrete samples and then inject it into the FlowCAM. I'm wondering if you've used it or it's possible to use with a flow-through system.
Yes, it is possible to use with a flow-through system. In fact, you can talk to the people at Fluid Imaging. They've had a site going for quite a long time. There are things that can happen on a moving boat, and like you can have the machine being twisted in the wind or whatever because of conditions, and so I like to make sure that there are no air bubbles in the sample, et cetera. In our case, we're going really, really fast along the river course. I really don't have time for the sample not to go through well, and so I put it in by hand to be sure it's good. In time, I'm hoping that we can adjust that and have it as a continuous flow. I'm just personally not ready, but I think it can do it all right.
Okay,
I do think it can.
This is Leah, and I have one more question. There are no other questions out there. Peggy, do you know, you have one that you can take on your research boat. So are they two different ones, one for the lab and one that you can take out, or is it pretty much the same and you take it out or use it in the lab as well? And are there any differences?
Right. We use the portable FlowCAM. We use that either in the field or in the lab. We have either option with that.
Okay.
The other one is a laboratory model, and that stays in the lab. So with us, we want the flexibility.
Okay. So and they both do the same thing. The
Correct.
one is in the lab is not better, so it's just probably a little more expensive to have a portable one. Okay, thank you.